Hello everyone, this is LATHA AKULA. In this video we are going to study about the next topic is polysaccharides. Already in the previous video we have completed what are monosaccharides, what are derived monosaccharides and oligosaccharides. The next one is the polysaccharides. So what is another name for the polysaccharides? They were also called as glycans. They were also called as glycans, and these are sweetless. They were not sweet in taste, and these are polymer or chain of monosaccharides. That means different monosaccharides, they were arranged one by one and form a chain of structure called as polysaccharides, and these were in the form of threads like cotton threads. They were in the form of threads and which contain different monosaccharides. Different number of monosaccharides were present in the chain of polysaccharide, and sometimes these polysaccharides they may be branched, and in some polysaccharides they may be unbranched. Branching may be seen in some, and sometimes they may be in the form of linear without any branches. And how these monosaccharides will be connected, different monosaccharides are present, right? How these monosaccharides they have been connected? They have been connected by means of glycosidic linkage or glycosidic bond. By means of glycosidic bond. So, and what are the different types of glycosidic bonds? Already we have studied about how the glycosidic bond is formed. How the glycosidic bond is formed? By the condensation of molecules. By the condensation of the different monosaccharides, they form the glycosidic bonds. For example, this is the alpha 1,4 linkage. This is beta 1,4 linkage. What is the difference between alpha and beta 1,4 linkage? Alpha 1,4 linkage, this is the alpha glucose. These are the alpha glucose. Alpha 1 glucose. And this is also alpha glucose. Alpha D glucose. This is beta D glucose. And this is also beta D glucose. Here, the differences between these two is in the first carbon, the configuration of hydrogen and hydroxyl as that changes. See here, in the alpha D glucose, what will happen? There is in the first carbon, hydrogen and hydroxyl ions will be there. And here also hydroxyl groups are there. Hydrogen and hydroxyl groups are there. So between these two, what will happen? Between the first carbon and fourth carbon, there is a dehydration takes place. That means a removal of water molecule. Removal of water molecule occurs, and the resulting one is oxygen. Only one molecule of oxygen is present. By this, there is a glycosidic linkage is formed. This is called alpha 1,4 linkage. And whereas in beta D glucose, the configuration of hydrogen and hydroxyl ions changes. So because of this, the hydroxyl ions comes this side. Here it is hydroxyl ion. So between these two, there is a linkage. How this occurs? Here, from here to here, hydrogen to hydroxyl ions, they will be removed in the form of water molecule. In the form of water molecule, and the remaining one is oxygen. With this, from here to here, there is a linkage occurs. This is called as beta 1,4 linkage. This is alpha 1,4 linkage occurs between the alpha D glucose, and the beta 1,4 linkage is formed between the beta D glucose, or it may be galactose or anything, any sugar. So we have studied that there is a branched structures and some of them have unbranched structures. And how these branched structures and unbranched structures they have been linked. This is the branched one. Here what we'll say, this is the linear form, this is the branching. And here in this branching, how these monomers have been connected by means of alpha 1,4 glycosidic linkages. Here the glycosidic linkage occurs between the first carbon to fourth carbon by the removal of water molecule. And sometimes some of the polysaccharides they have branching. The branching arises because of the linkage of sixth carbon with first carbon of another monosaccharide. Here from the sixth carbon to first carbon, if there is a linkage occurs, glycosidic linkage occurs, that is called as alpha 1,6 linkage. Alpha 1,6 glycosidic linkage. And here we need to know alpha 1,4 glycosidic linkage occurs in the linear polysaccharides, and in the branching structures, what type of the linkage we need to remember? Alpha 1,6 linkage. Alpha 1,6 linkage. Like this, some of the polysaccharides, they may exhibit branching pattern. And depending upon the composition, the polysaccharides are of two types. They were Homo polysaccharides and Hetero Polysaccharides. Homo polysaccharides, they consist of only one type of monomers or monosaccharides. Only one type of monomers or monosaccharides. They were arranged one by one and condensed to form the glycosidic linkage and form a chain of structures that is called as Homo polysaccharides. And heteropolysaccharides means more than one type of monosaccharides. More than one type of monosaccharides, they have formed the polysaccharide that is called as heteropolysaccharide. Next we will see what are Homo polysaccharides. In this video we will study about Homo polysaccharides. So actually polysaccharides are of two types based on chemical composition. They were Homo polysaccharides and Heteropolysaccharides. Homo polysaccharides were made up of one type of monosaccharide units, and heteropolysaccharides were made up of more than one type of monosaccharide units, right? So in this video we will study about some examples of Homo polysaccharides. So that the first one is glycogen. So glycogen is commonly called as animal starch. In animals, excess carbohydrates were stored in the form of glycogen in the liver muscles, and from that liver and muscles, this glycogen, they release energy in mammals. And here if you see one molecule of glycogen, it consists of nearly 30,000 glucose units. And here this is the glycogen structure. This unbranched unit, it has alpha 1,4 linkage, and this branched unit have alpha 1,6 linkage, as we already seen in the previous video. When the glycogen is treated with iodine, it gives a characteristic red color. And second one is the starch. So like glycogen, which is the storage polysaccharide, starch is also one of the storage polysaccharide. Here it is present in the plants. The glycogen is present in the animals and starch is present in the plants. In the plants also, the excess carbohydrates were stored in the form of starch. And here this storage polysaccharide, it will be in the form of helical secondary structure, which will fold like helix. See, this is the helical structure. And when it is treated with iodine, it gives blue color. Here the glycogen, what is the color given by the glycogen when it is treated with iodine? It is red. Whereas the starch gives blue color. And where the starch is present in the plants, it is present in rice, wheat, tubers, potato, tapioca, apple, banana. So in all these, starch is present. And starch is in two forms. One is amylose form and second one is amylopectin form. The amylose is unbranched structure, whereas amylopectin is a branched structure. See, here from this one branching is arising, right? So amylopectin is a branched structure, whereas amylose is an unbranched form. And here in unbranched form, what is the linkage present generally? Alpha 1,4 linkage. Whereas in the branched form, in the linear form, there is alpha 1,4 linkage. Wherever the branching was arises, there is alpha 1,6 linkage will exist. And here one molecule of amylose consists of nearly 200 to 1000 glucose units, whereas one molecule of amylopectin, it contains 2000 to 2 lakhs glucose units. And the next one is cellulose. Cellulose is the structural element in plants. Here structural elements means what? This is cellulose, it forms the structure of plant cell wall. Hence it is called as structural polysaccharide. And where it is present, we are seeing paper, right? The paper is made up of cellulose. Cotton is also made up of cellulose only. Paper, cotton fibers, they consist of cellulose only. And these are unbranched form. How the cellulose exists? It is in unbranched form. If we see the one cellulose molecule, it consists of nearly 6000 beta glucose units. And if you see cotton fibers, they have large amount of cellulose, that is nearly 90% of cellulose is present in the cotton fibers. Whereas wood Wood consists of nearly 25 to 50% of cellulose is present, and the remaining amount will be lignin and pectin. And here if you see cotton fibers and wood, they consist of abundant amount of cellulose. Largest amount of cellulose is present in cotton fibers and wood. And artificial fiber, that is nothing but rayon. Rayon is also prepared from the cellulose only. When the cellulose is treated with alkali and by performing some other functions, the rayon will be obtained from the cellulose. And here, if the cellulose is treated with some chemicals, like they form cellulose acetate. If it is treated with some chemicals that form the cellulose acetate, this cellulose acetate is used in fabrics, making the cellulose plastics, and also used for the making the shatterproof glass. And another one, cellulose nitrate. This cellulose nitrate is used in explosions. In the explosions, it is used as a propellant. And carboxymethyl cellulose is used as an emulsifier in making the ice creams, cosmetics, medicines. So this carboxymethyl cellulose that gives smooth texture to these ice creams and cosmetics and medicines. And cellulose is not digested by the human beings because cellulose is a complex carbohydrate. For digesting that cellulose, we need an enzyme called as cellulase. So in our body, there is no cellulose enzyme to digest it into the simple glucose units. But who will digest it? So if you see in the body of, we know about ruminants. Ruminants means the cattle which are grazing on the grass, right? So in those cattle, they were called as ruminants. They consist of extra digestive pouch which is called as a rumen. In the rumen, there are some microorganisms, some bacteria were present which will release enzyme cellulase to digest the cellulose into the simple sugars. So only the rumens can digest the cellulose. Next one is cellulose is the most abundant carbohydrate in biosphere. So cellulose is the most abundant carbohydrate in the biosphere, and cellulose is produced by the plants in making the cell wall. So this is about cellulose. Next Homo polysaccharide is inulin. Inulin is another Homo polysaccharide. It is a polymer of fructose. That means it is made up of several units of fructose, and it is a storage polysaccharide. So like starch, it is also a storage polysaccharide where it is present in the roots and tubers of dahlia and related plants. Dahlia related plants, the roots and tubers, they consist of this inulin, and it is not metabolized by the human body. That means if we consume those tubers, that was not properly digested, that was not properly metabolized by our body, that means that inulin will be removed by the, filtered by the kidneys. Hence, this inulin is used to detect the proper functioning of the kidney also. And so, this is important one. Which polysaccharide is used to test the kidney functioning means we need to mention inulin. And next one is chitin. Chitin is the second most abundant organic substance on the biosphere. What is the first abundant component? That is cellulose. And the second abundant component is chitin. Where it is present? It is found in fungi and bacterial cell wall. The fungal and bacterial cell wall, they consist of chitin, and in this chitin, the basic unit is nitrogen containing glucose derivative, that is N-acetyl glucose amine. In shortcut, it is called as NAG. And N-acetyl glucose amine. N-acetyl glucosamine, and this chitin is also present in the exoskeleton of arthropods, that is insects. Exoskeleton also is made up of chitin only. And with this chitin, we have completed Homo polysaccharide. The next one is Hetero Polysaccharide, the last topic in the polysaccharide, the last topic in the carbohydrates. So Hetero Polysaccharides, they were made up of more than one type of monomers or more than one type of monosaccharides, and it is complex because it is made up of more than one monosaccharide, hence it is called as complex polysaccharide. And in this we have two examples. One is peptidoglycan and another one is a hyaluronic acid. That the important example is the peptidoglycan. So we have already heard this word peptidoglycan while studying the bacterial cell wall, right? Here this peptidoglycan is present in the cell wall of bacteria, and it contains chains of two alternative units of amino sugars molecules like N-acetyl glucosamine and N-acetyl muramic acid. N-acetyl glucosamine shortcut is NAG. N-acetyl muramic acid shortcut is NAM. Alternate subunits of NAM, NAG were linked by means of a glycosidic linkage, glycosidic bond. Here, the lysozyme is the enzyme that hydrolyzes this linkage. Here, where this lysozyme is present in our body, the lysozyme is present in our tears. In our tears, this lysozyme enzyme will act as a defense mechanism against bacterial infections to the eyes. And the last one is hyaluronic acid. It is present in the it gives a flexibility to the tendons and cartilage, and it consists of D-glucouronic acid and D-N-acetyl glucosamine. This D-glucouronic acid is a carboxylic acid. D-N-acetyl glucosamine, it is a derivative of the glucose units. So this hyaluronic acid is made up of these two units, which will give the flexibility to the cartilage and tendon. And with this we have completed the polysaccharides, we have completed the total carbohydrates, and in the next video we will start with the amino acids.